Hello, everyone. My name is Dylan Barth, and I'm here today to talk to you about the enhancer interactome and its effect on gene regulation. In order to understand the basis of gene regulation, we attempt to characterize the properties of functional enhancers through a machine learning approach. We build off of an existing model, the activity by contact model, which only has two features of enhancers: activity, which is comprised of H3K27AC and DNase hypersensitivity, and contact, which is measured by high C with the promoter of the gene. This model leaves out the role of potential enhancer-enhancer contact, causing indirect regulation of the gene. To address this, we characterize enhancers as either directly, E1, or indirectly, E2, E3, etc., contacting the gene. Then, by utilizing a CRISPR-I dataset from Gasparini et al., which tests if blocking enhancers affects gene expression, we construct a machine learning model that predicts the importance of enhancers to a gene based on their characterization. We also test the importance of various ChIP-seq features in predicting functional enhancers. This is the contact network of the JunB gene. Here, the red square shows the promoter, and each of these circles is an enhancer within 5 megabases of that promoter. The activity level of each enhancer is shown as the color, where blue is very low activity and green is high activity. Each edge represents the high C contact between that enhancer and another enhancer or the promoter. Edges are weighted higher when the high C contact is more, and they are red when they connect an enhancer to the promoter. You can see here that there are a few enhancers that connect directly to the promoter, but many of the connections happen between enhancers. These are the connections that we're interested in this study. So now let's take a look at the methods. Basically, what we start with is predicting enhancer-promoter pairs using the activity by contact software. Then we create a contact network like the one we saw above from the high C data. From that network, we classify the enhancers based on their contact as either E1, which is a direct connection, or indirect connections as E2 and E3. Then we extract the ChIP-seq data from ONCODE and select CRISPR-I validated enhancers for our training and test set. Finally, we train the machine learning algorithms and select out the most important features of functional enhancers to determine what the features are that affect an enhancer the most. Looking at our results, let's just take a look at the significant versus non-significant enhancers first. Out of about 66,000 enhancer gene pairs that were tested, only about 600 were found to significantly alter expression of their constituent gene. Of those 605 significant EG pairs, 382 were E1, 36 were E2, 11 were E3, and the remaining 176 were classified as EINF, which means they either were more than 3 steps out from the promoter or they had no significant contact within the network at all. Enhancer roles affirm the ABC model's predictions. You can see that as E1, E2, and E3 progresses, the high C contact with the promoter, especially in the significant or functional enhancer column here, goes down. This makes sense because we would expect if it's a further step away in the network, it has much less contact with the promoter. So that's like what an E3 is. And you can see that the E3s that are functional enhancers have to make up for that lack of contact with a little bit more activity. If they're further away or for some reason come into contact with the promoter of the gene less, they need to compensate for that a little bit by recruiting transcription factors and other molecular processes to that enhancer. Once we've trained the models, the performance of each model can be seen on this precision recall curve. We optimize based on the F1 score because there's a high amount of true negatives. You can see that the XG boost is the best performing model by far, and ABC model, even though it only contained 2 variables, still performed better than some of our models with way more. Also, the linear distance between the enhancer and the promoter is a very bad predictor. So from the best model here, the XG boost model, we pulled out the top 20 features, and that's shown in this graph here. Obviously, contact and activity are really important features. We already knew that from the ABC model. Some other interesting things here show transcription factors that you might expect in K562, which is the cell line we're working in. GATA1, STAT5A, these are pretty classic K562A transcription factors. Another interesting one was CTCF in the enhancer was an important feature, showing that if you disrupt a chromatin loop, that can, in fact, perturb gene expression. So lastly, we trained those same models on subsets of the data. One model was trained on only E1s, and then the other model was trained with E2 and above, so E2, E3, EI, and F. We took the feature importances from those models and we subtracted them from one another. So this graph shows the importance difference between those E2 and E1 models. So down here, we see high importance difference between them. That means that those features were much more important when we trained only on indirect connections than they were when we trained on direct connections to the gene. You can see a lot of repressive marks that are really important in E2 that were less important in E1. This may lead to the conclusion that E2s are repressive elements. However, in the Gasparini dataset, it was only called a significant enhancer if it downregulated the expression of the gene when it was blocked. That means that these aren't repressive elements because when they were blocked, the gene expression actually went down. This is a bit of a mystery, but there are some interesting results showing repressive complexes actually being used in a positive manner. This may point to some of those similar results. In conclusion, we developed a novel method for predicting the importance of enhancers to nearby genes using machine learning on 3D genome structure and other genomics data. And we also underscored the importance of enhancer-enhancer interactions and their role in gene expression. Thank you guys for watching today, and I'd love to take any questions that you have in person.